Uh, okay, can you guys see? All right. Great. So yeah, thank you for Open Box Science for organizing this. It's always really exciting to be able to give these talks to a really sort of broad audience from all over the place. You know, really thank you for organizing this and bringing all the different speakers together on this topic. So today I'm going to talk about predominantly the work that I did during my time as a graduate student in Jeff Friedman's lab. And, you know, I'm fortunate I get to go after Jessica because she gave, you know, a really beautiful introduction and sort of the interesting parts of sickness and what we're sort of, why we're sort of interested in sort of studying this. So I'm not going to spend too much time giving the same background again, but I'll just reiterate some points to sort of orient you to how we're thinking about sickness. So, you know, usually, you know, we're all excited to go to lab, you know, hang out with our lab mates, grab lunch together, maybe do a couple of experiments, you know, during the day. But this is something that we're now sort of hyper aware of with COVID. Once in a while, we start to feel sick. And, you know, what does that mean? Means that all of a sudden, we're not really interested in hanging out with our friends. We're not hungry or thirsty as much anymore. We might have a fever. And all of a sudden, our bed seems like the most interesting place in the world to be. And these really stereotype changes in behavior, you know, happen across all humans, but it's also been recognized that this is a sort of remarkably conserved set of changes across basically all animals. And it was in 1988 that this veterinarian, Benjamin Hart, put forth this idea that these changes that happen during infections in humans and in animals are not just due to damage by, you know, whatever pathogen or infection we have, the bacteria or virus, but it's actually something that's induced by our bodies itself to help fight and survive infections. So since then, there's been a lot of really, you know, nice work showing that this is indeed the case. And here I'm going to point out one, I think, really convincing example of this, where if you take animals that are sick, that are usually anorexics and they're not eating much, and you force feed them, so this is during a bacterial infection, you actually see that animals succumb to infection and die more. So this really indicates that these changes are a really important sort of control mechanism in our body for being able to better survive infection. So I'm not going to go into, since then, a lot of the other work that's been done, and I think Jessica, you know, touched on a lot of these things about how the immune system, you know, is the sort of the sensory system that recognizes infection, converts that into these sort of circulating signals called cytokines, such as, you know, IL-1, TNF-alpha, as well as others. And this information has been shown to be able to make it to the brain through circulation, as well as peripheral afferent systems like the vagus nerve and spinal afferent systems, to induce the behavioral changes that we're talking about. What was less clear at the time was sort of how are these changes coordinated in the brain, and what populations of neurons in the brain might be really important for mediating these changes. So that was sort of the goal, is to identify a neural substrate for sickness behaviors. And the work that I'm going to talk about today was you know, really profoundly helped by another graduate student in the lab, Rainy Xiao, and a phenomenal research assistant, Jordan Shakin. So when we wanted to look at these changes in behavior, our focus was really on sort of more homeostatically regulated behaviors, such as food intake, water intake, and movements. So what we wanted was a really effective home cage monitoring system for these different behaviors that allowed us to have really high resolution tracking, but tracking over days and weeks, which is sort of the more relevant timescale for sickness. So what we ended up having is sort of a home cage setup where animals can basically, get food through a pellet dispenser that monitors every time they get food. We have overhead video camera recordings, which give us sort of the locomotion, but also allow us to extract other behavioral features that I probably won't talk about, as well as a lichometer to sort of get the water intake. So this allowed us to really profile the behavioral changes that we care about a lot in really good detail. So with this system in place, now we could start to ask the question of what does sickness behavior look like? And again, we use the same sort of model that Jessica talked about, which is an injection of LPS. And LPS, again, is a component of gram-negative bacteria that is really well studied from the immune side to be a key sort of molecule that our immune system recognized to sort of say that we have a bacterial infection. And there's an added advantage to using LPS, and that is that since this isn't a live infection, there's actually no damage being induced in the body through the course of this. So this is really just a way to induce a bacterial immune response. And what I'd like to point out here is that we tried multiple different doses of LPS, and this is actually higher doses than what Jessica used in her work. So this is important to keep in mind for sort of this scale of the behavior and even some dichotomy in behavior. So here are sort of example traces, and this is how a lot of the data will be shown for the food intake, water intake, and movement, as well as a trace of the movement for 24 hours in sort of saline treated animals. So this is baseline behavior over 72 hours in this case. And then if we look at what happens when we give these different doses of LPS, we could see that all of the doses produce sort of a really remarkable shift in the behavior of animals. And what we recognized is that just like other sensory systems that sort of provide information to the brain, that the response scales with dose, where you could see that the lowest dose has a weaker and shorter lived response than our highest dose. And that we see that amongst the doses, we see a sort of coordinated induction and duration of the response. So the food intake, water intake, and movement changes that we see are fairly coordinated amongst all of the doses, indicating that this is sort of a behavioral program that's induced. And we also looked at autonomic changes such as brown adipose tissue temperature changes, as well as core body temperature changes. And what we see at these doses, especially since we're not providing any sort of bedding or ways for animals to generate heat effectively, they actually have a more hypothermic response, which has been recognized in mice due to their smaller body size and stuff as a response that happens during infection as well. So in this case, it's a little different in terms of the thermic response than what Jessica was studying, but a lot of the other sickness behaviors are sort of pretty well maintained. So with this sort of system established and phenotyped, what we then wanted to move on to was understanding what's the sort of activity in the brain during infection. So we again used staining for this immediate early gene called FOS, which is a pretty good proxy for neuronal activity. And we did this using whole mount tissue clearing and staining, which allows us to sort of keep the entire brain intact and look brain-wide at the response. So this is an example brain of an animal injected with 0.5 mg per kg of LPS, which is the dose that we stuck with for the remainder of the paper at 3 hours post-injection. What you could see is that there are many nuclei throughout the brain that have pretty profound increases in the amount of FOS. So each of those dots is an active cell in the brain. And the first takeaway from this is that the changes caused by LPS in the brain are pretty widespread. It's not just one or two regions, but this seems to be a really like systems level response. And we could, further quantify this, especially over, a time course. And what we see is that The response peaks at around 3 to 6 hours, and again, that there's quite a lot of activity for animals that are basically not doing much. So this tells us that there's a lot of information sort of being conveyed to the brain and going on. And one set of regions that we were really excited by was the nucleus tractus solitari and the area postrema, as they showed early and really strong sustained activation during sickness. And beyond that, the reason why we were excited by this brainstem region, so this is the brainstem, cerebellum, and this is the region that we specifically look at, is that it's a blood-brain barrier permeable region. Again, Jessica mentioned this for CBOs. So circulating factors actually can directly interface with the neurons and cells of this region. And importantly, it's also the entry point for the afferent vagus nerve. So this seemed like a really important sensory hub for interoception in the body, making it a really ideal candidate for further follow-up. And finally, there's rich literature on an abundance of different energy balance related to receptors and neuropeptides, which made sense for homeostatic behavioral control. So to follow up on this, we also use the TRAP2 mouse line, which is a really effective way to basically induce whatever genes that you would like in specific populations of activated cells. And again, this is done by a Cre-ER that is under the control of the FOS promoter, meaning that whenever there's FOS expression, there's this Cre-ER that's produced. And then we can give tamoxifen to animals to basically functionalize this Cre for 4 to 6 hours. So when we have a specific state, such as sickness, we can then give tamoxifen and basically label. And in our case, what we wanted to do was manipulate neurons that were activated usually by LPS. So we use chemogenetics and specifically, DREADS to be able to activate and inhibit neurons of choice. And our basic sort of paradigm is that we can inject the virus into just the area postrema MTS region. So we have really localized injection of Cre-dependent DREADS, and then we can either label mice that are treated with saline, so there's not going to be much labeling in this condition for this region, or in mice injected with LPS, where there's a lot of activity in this region. We could let the animals then recover from the LPS, and now that these cells the activated cells are labeled, we can ask if we reactivate these cells at a later time point, what happens? And what we find is that there's a really sort of remarkable decrease in this case, what I'm showing right now is food intake, but this also holds true for water intake, movement, and temperature changes, as well as body weight, indicating that the population of active neurons in this region is really able to produce sort of this potent, long-lasting sickness behavior that is characteristic of LPS. Now that we had a region in hand, we wanted to get a better idea of sort of which cells in this region are actually the activated ones, and can we get a better understanding of sort of the segregation and sort of the logic of behavioral control from this region. So to start, we did some baseline sequencing to just get an atlas of cells in this region. And then what we did is we labeled the activated cells with a nuclear GFP, again, using the TRAP2 line. And we specifically isolated those cells and sequenced those cells to see which are the activated cell types in this region. So this is just an overview of sort of the baseline cell types in this region. There's a diversity of different cell types, excitatory and inhibitory. And then when we look specifically at the cells that we isolated from when we did activity sort of based sequencing, we find that that it's not just a single cell type in this region that's activated. We see inhibitory cells as well as excitatory cells activated. But what we did see is that there are specific subpopulations of activated cells according to our sequencing. And I'm not going to present the data here, but we decided to focus on excitatory cells because we saw that this effect was predominantly driven by excitatory cells. So within excitatory cells, we looked at which sort of cell type markers were the most representative of the activated neurons, and we found that the best enriched cell type was ADCYAP1 neurons. And this was enriched in the NTS and the area postroma region. But it's important to note that it wasn't the only cell type that was activated. We did see that other cell types, which have overlapping expression patterns, were also enriched. So we first wanted to check what the role of ADCYAP1 neurons were. So we used a similar approach where we injected pre-dependent activating dreads into this region in ADCYAP1 Cree mice. So just ADCYAP1 neurons can be reactivated. And when we did that, what we found is reactivation of just this one neuronal type is able to produce really profound changes in food intake, water intake, movement, reductions in temperature, and body weight. So this indicated to us that this population really recapitulates the sickness phenomenon. And this was remarkably close to what we saw with the TRAP2 line that just labeled all of the active cells in this region. So this really indicated that this is most likely the key cell type in this region. But we went on to sort of test basically all the cell types that we thought had some level of enrichment during this phenomenon to see if there's any sort of specific behavioral dissociation or logic. And what we found is that generally there's no dissociation of food intake, water intake, and movement among cell types in this region. And that ADCYAP1 neurons really had the strongest phenotype of the ones tested. While we were able to show that these neurons were sufficient, we now needed, while that shows they are capable of inducing the sickness response, what we next wanted to test is whether these neurons are actually required for the sickness response that's induced during LPS. So here the logic is we want to inactivate these neurons, specifically give the animals LPS and see what happens. So this is a little more confusing, but basically here in gray, what I'm showing you is animals that have just M-cherry expression in these neurons. So, they can't be inactivated. And if we just give them LPS and CNO, you could see that there's a really strong reduction in food intake, just basically characteristic of just standard LPS injected mice. In yellow is sort of our other control of this is sort of like a baseline animal. So the GI dreads animals where they're injected with just saline to give us an idea of what just normal animals eat. And then here in this brown, what we see is this is what happens when we inactivate the ADCYAP1 neurons and give LPS. And what you see is that it's able to rescue around half of the food intake difference that we see if we just give animals LPS. But it's important to note that it doesn't fully rescue this phenomenon, that it doesn't just bring animals back to a baseline sort of state. So this indicates that while these neurons are important for sort of the full magnitude of the sickness response, that there are most likely other neuronal types throughout the brain contributing to control of food intake, water intake, and movement, as we saw that this was similar across all of them, where we can rescue about only half of the effect, despite being able to sort of induce the entire effect. And then we saw actually that this was not able to ablate the hypothermic response, indicating that there are probably other important thermal control mechanisms, and that this was able to abate some of the body weight change. So this gave us confidence that these neurons are indeed involved in the usual generation of sickness. And when we test other lines that were also, enriched during during LPS injection in this region, such as DBH neurons or FLAX2B Cree neurons, we find that inactivating these neurons isn't able to really change the sickness response that we see, indicating that the ADCYAP1 positive neurons are this really important subset for regulation of sickness behaviors in this region. So to sort of summarize, our goal at the onset of this project was to identify an important population for control of sickness behaviors. And that's what we think we found with ADCYAP1 neurons. But this is really just the first step and sort of just a handle into looking at how the brain actually controls sickness behaviors. So this really opens up avenues for very important questions, such as how are these neurons actually being activated? So I brought up that the peripheral nervous system is able to transduce signals produced by the immune system to the brain, and that this region of the brain is actually the site that all vagal information comes into the brain. So it will be really important to see if the vagus nerve sort of activates these neurons, as well as looking at humoral signals, as a lot of signals produced by the immune system circulate in the blood, and that this region has access to that. So in the future, and this is work that's ongoing, we're trying to figure out how these neurons are activated. We also showed that there are a ton of other regions that are activated in the brain during LPS-induced sickness. So what are these other regions contributing, and can we have sort of a more holistic idea of sort of the connection of these regions and how this is sort of hierarchically organized? And then finally, what is the effector circuit responsible sort of for the output of these changes? We showed that we can get sort of a coordinated change in this region, but maybe if we look downstream, we could see sort of better dissociation, the way that Jessica sees for some of the responses that she looked at, where we could see sort of more dedicated populations for different aspects of the behavioral control, and then maybe there we could sort of just manipulate those and change that behavior really potently. So, well, this has sort of been the initial focus of trying to understand how the immune system sort of communicates with the brain to affect this important aspect of our body, which is behavior. It's important to recognize that during sickness, there are also changes in other aspects of our body, such as autonomic functions or organ function and metabolism. And the overall goal really is to try to understand this axis from the point of view of what immune signals are produced that are actually relevant to the brain, what are they signaling to the brain, and how is the brain sort of processing this information? And finally, how is it leading to a change in sort of coordinated physiology amongst behavior, autonomic function, metabolism, and how is that really important for survival during infection as well as regulation of infection? So I'm going to touch on a little bit of work that we're doing to sort of establish this as sort of the next focus. Well, I've told you, we really focused on setting up a good behavioral system to look at the sickness behaviors. For these other aspects of physiology, we want to do the same thing. So for autonomic function, this is work that I've been doing with clinical fellow Karthik and research assistant Jordan. And we've been using pulse oximetry and telemetry to look at sort of vital sign function in the form of heart rate, breath rate, temperature, and pulse distension. And what we see is at higher doses of LPS, and that there are actually profound changes in these vital sign functions indicative of a whole body sort of response to sort of maintaining a different physiological state. And in the future, we'd really like to understand how these different responses are controlled, and coordinated with that are metabolic changes in the body, because we see that animals actually shift what fuel sources they use. So here I'm showing you respirometry data, which measures CO2 production versus O2 production. From that, we can tell what sorts of fuel the animal's using. And usually animals have a respiratory quotient closer to 0.9 or maybe even 1, indicative that animals are using primarily carbohydrate-based fuels. But when we see this sort of drop in the respiratory quotient, this is indicative to us that animals have actually shifted towards using lipids instead of carbohydrates. And then we can also do more snapshot-based profiling of blood, and in this case metabolomics, to show which specific metabolites are changed. And as you can see, there's really profound changes that happen during infection, during the metabolic profile of animals. So this all goes together to really influence the entire physiological state during infection. So by trying to study all of it in a sort of coordinated manner, I think we could get a better understanding of how the neuroimmune axis functions during infection to coordinate entire bodily function. But hopefully by using this sort of logic, we can also pull out general principles for how the immune system and the brain coordinate with one another. And we can hopefully generalize this to other immune relevant states such as stress, cancer, and toxins. So with that, I'd like to thank my graduate mentor, Jeffrey Friedman, who's honestly been an incredibly supportive and generous mentor, and who's just incredibly gifted in working on sort of homeostatic control problems and energy balance, as well as Rainey, Jordan, and Karthik, who I mentioned throughout the talk, have really helped in really critical parts of this project. And I'm now, starting my lab at Janelia, and we have a grad student, and we're looking for postdocs now, as well as research assistants and research specialists to really, move some of these projects forward. And then finally, I'd like to just say that next week, there's actually a really great set of virtual talks in this 4D cellular physiology symposium held by Jennifer and Ron here, talking about, brain-body interactions. And I think, Vivian's mentor Ruslan's going to be giving a talk. Chiu's mentor Steve is also going to be giving a talk, as well as many other people across, immunology, cellular physiology, and brain-body interaction. So I encourage you to sign up for that. So, take any questions.
Thank you very much, Anoj. One question that I have for you before we move on to a couple of different questions from the audience is that, what do you think about, so you give LPS to mice, they have the sickness behavior, and I assume it dies down after a few days. Once these animals are challenged again with LPS, do you think it's going to be different from mice that haven't been sick yet?
Yeah. So we were really careful about this. The only experiment where mice were injected twice with LPS was the TRAP2 experiment, because inherently we had to first label them to be able to do a second round when we inactivate them. But we do see that there's actually a difference in the response where the response becomes weaker with subsequent injections of LPS. So naive responses are actually sort of the strongest, and after that they become weaker and weaker. And there's interesting dynamics to what happens depending on how far out you space the two doses, what dose you use, and such. So it's almost like a tolerance mechanism to this. But yeah, good question.
Cool. So I'll take a question from the audience. Have you checked the effect of neuronal activity upon LPS stimulation in microglia depleted mice?
Sorry, what was that? In
microglia depleted mice, have you checked the effect of neuronal activity on LPS stimulation?
No, and I think this is a really good point, sort of at trying to get at sort of whole brain, or just sort of what's happening in the CNS in terms of what signals are being directly, transmitted to the neurons, and which signals are actually going through glial cells such as microglia and stuff. So we haven't looked at that, and I think this is a really important sort of next step in trying to get at the mechanisms of induction and sort of the global state of maintenance that we're really interested in. And actually, I didn't talk about this, but this is we're working closely with a lab at Janelia that also works in zebrafish, so we can sort of do whole brain imaging of glial cells and neurons when animals are sick. So this is ongoing collaborative work on that front, but no clear idea yet.
Awesome. Take one more question, I think. What are the differences and mechanisms that result in the differences of low dose LPS to high dose LPS-induced sickness?
Yes, I think this is a really important point where I think that one of the things is at really, so at really high doses, you start to see much stronger changes. So we don't see these changes at the doses that we used at like 0.5 mg per kg, where now at higher doses, you see changes in heart rate, breath rate, and pulse distension, which are, approaching conditions similar to more septic shock. So this is really important for a really just potent immune stimuli with a sort of cytokine storm. And we're really interested in this phenomenon as well, and sort of what is different about this sort of shift where now there's a complete change in sort of organ physiology as compared to lower doses, where I think that there, and amongst these doses, there are differences, even at like sort of lower doses. I think a lot of that's probably driven by how much cytokine is produced and sort of the duration of the immune response to clearance of LPS. So I think that really affects sort of the time scale and sort of the propagation of the effect. So I would say that it's most likely due to sort of the potency of the cytokines produced in the initial response, which is a pro-inflammatory response. But I actually think an important part of this is the sort of the counter-regulatory anti-inflammatory response that's induced after, and sort of the magnitude and balance of those that kind of determines the differences in mechanism. But yeah, I think that, we'll still need to sort of figure some of these things out, especially when it comes to physiology, by looking at different doses and getting a better idea of how the whole body responds rather than just looking at behavior.
Awesome. Thank you very much for the beautiful talk and for answering all our questions. Vivian, hand